what if there was a poison hiding on your dinner plate? In this explainer, we're essentially opening up a scientific case file. We're going to follow a team of toxicologists as they hunt for a hidden danger in our food. Just think about it for a second. Your favorite seafood dinner. What if it was harboring a toxic heavy metal and you'd have no idea? That's pretty much the exact question that kicks off the whole investigation we're diving into today. Okay, so here are the specifics of the case. We've got a team of toxicologists, and they have this hunch that seafood from two major U.S. coastal areas might have different levels of contamination. So they're zeroing in on the Gulf Coast, we'll call that Region A, and the Pacific Coast, we'll call that Region B.
All right, so a poison on the plate. What exactly are they looking for here?
The target is cadmium. It's a heavy metal and it is highly toxic to us. The real danger isn't just getting sick right away. No, it's that cadmium builds up in our bodies over time, accumulating in vital organs like the kidneys and liver. And that can eventually cause some really serious long-term damage. So, to get to the bottom of this mystery, the investigators can't just, test a random piece of fish. It's not that simple. First, they have to prepare the evidence with extreme care to make sure their readings are going to be perfectly accurate. It's this incredibly meticulous four-step process. So after collecting the fish and human tissue samples, they're homogenized, which is basically just a fancy word for being blended into a perfectly uniform paste. Then they use these powerful acids to completely dissolve the tissue, which releases all the elements that were locked inside. Finally, it's all carefully diluted. Every single step here is designed to create a clean, consistent sample and prevent any outside contamination from messing up the evidence. Okay, so with the evidence all prepped and ready to go, the team needs a special tool. They need a machine that's powerful enough to find a needle in a haystack, or in this case, a single toxic atom in a whole sea of biological material. Their tool of choice is a technology called ICP-MS. Think of it as the ultimate elemental detector. This thing is so incredibly sensitive, it can find even the tiniest trace of an element, like our cadmium, hidden away inside that complex sample of digested tissue. And this really highlights why the scientists chose ICP-MS. See, older techniques like atomic absorption, they're cheaper, sure, but they're also slow, and they can only measure one element at a time. ICP-MS, though, even with the higher cost, is a total game changer. It's way faster, far more sensitive, and it can screen for a whole suite of elements all at once, making it the perfect tool for a complex investigation like this one. Okay, so we've talked up this super detector, but how exactly does it work its magic? Well, the sample goes on this incredible four-stage journey. First, it gets turned into a fine mist, kind of like a perfume spray. Then, that mist is shot into a superheated plasma that just atomizes and ionizes everything. A mass filter then sorts these brand new electrically charged atoms by their specific weight. And finally, a detector at the end counts up each type of atom one by one. And when we say superheated, we really mean it. The argon plasma that atomizes the sample can reach a staggering temperature of up to 10,000 Kelvin. To put that in perspective for you, that's hotter than the surface of the sun. So that's the crucial point right there. That extreme heat is what makes the whole analysis possible. It completely obliterates the original sample, breaking it down into its fundamental atomic building blocks. This allows the mass analyzer to act like a super precise filter, sorting each and every element based on its unique atomic weight. So the machine gives them a raw signal, but that's not the final answer. How did the scientists translate that data into a concrete measurement of the contamination? Right? The machine doesn't just give a final answer. It provides a raw signal intensity that's measured in counts per second. That's basically just how many cadmium atoms it detects every single second. So the investigators' next job is to translate that raw signal into a meaningful concentration. They do this using something called a calibration curve. They'll run samples with known amounts of cadmium, let's say 0.5, 1, and 2 parts per million, and then they plot the machine's signal for each one. This creates a reference graph, a kind of scientific ruler, that allows them to take the signal from their unknown fish sample and pinpoint its exact cadmium concentration. After all of that meticulous preparation and some seriously high-tech analysis, the results are finally in. So, what did the scientific detectives find? The first major clue comes from the fish, and the data is pretty stark. As you can see here, fish from region A, the Gulf Coast, had an average cadmium level of 3 parts per million. That means for every million parts of fish tissue, 3 were cadmium. Now, that was significantly higher than the 1 part per million found in fish from region B, the Pacific Coast. Yeah, the numbers don't lie. When the team averaged all the results, they found a 3-fold difference in contamination. This confirms that where the seafood comes from really matters. And of course, this raises the next critical question for the investigators. Is this difference because of industrial pollution, or is it maybe because of natural geological variations in those regions? And here is the verdict that truly matters for public health. When they analyzed the human liver tissue sample, they found a cadmium level of 2.5 parts per million. Now, to put that in perspective, the safe limit set by the World Health Organization is less than 0.5 ppm. The level they found was 5 times higher than that accepted safety threshold. You know, this case is such a powerful reminder of how advanced science can make invisible dangers visible. Technologies like ICP-MS are absolutely essential tools for protecting public health. And it leaves us with a final crucial question, doesn't it? What other invisible threats are just waiting to be uncovered in the food we eat and the world all around us?